going to present our last results from the experiment to investigate certain utilization properties of novel composite material for developing cardiovascular implants. The role of endothelial progenitor cells. Cardiovascular implants are the most commonly used biomedical devices, such as heart valves, bypass grafts, and stents. And however, their application is associated with major complications. In the case of heart valve, for example, for mechanical heart valves, we have increased risk of thrombosis and thromboembolism. And for tissue or bioprosthetic heart valves, they are suffering from this durability and need to reoperation. One solution can be the use of advanced biomaterials, and we have developed new nanocomposite material based on polyethylene and polycarbonate polyurethane with improved properties for this kind of application. This is a schematic diagram of the structural post-PC nanocomposite. You can see the post nanocage incorporated into the backbone of the polymer as a chain. The aim of this study was to investigate to isolate endothelial progenitor cells from peripheral blood and then to culture nanocomposite samples to investigate their proliferation and differentiation for the endothelial cells. Why we chose endothelial progenitor cells? Because they are cells with proliferative capacity and they can differentiate endothelial cells. They can be found in circulating blood and endothelialization finally can offer us antithrombogenic surface, immune reaction, and protective valve and other implants. We can recognize EPCs using some specific cell surface markers, such as CD133, CD24, and EGFR that we use in our experiment. We isolated EPCs using MAX technique or magnetic activated cell sorting system. Then we characterize them using fluorescent activated cell sorting microscopic examination. We cultured EPCs on different polymers on our nanocomposite, and some polymers, surface modified polymers with RGD peptide and RGD conjugated lauric acid peptide as controls. And we use specific material cell. Here is a PAX plot. You can see the purification that we achieved after isolation of EPC. At the above shows the labeled cells, and underneath is unlabeled cells that shows just no CD34 positive cells. We compared, we used two sources, peripheral blood and umbilical cord blood, to isolate EPCs, and we compared the results that we get from the two sources. In terms of frequency of isolated EPCs, as we expected, the cord blood was more frequent, the EPC compared to peripheral. And we were interested in to see EPC expansion. We used triple positive EPCs, positive for CD34, CD133, and EGFR that are considered as true EPCs. And as you can see, over the time, the expression of triple positive PCs, or the frequency has increased, showing that we have an expansion in these subsets. In terms of differentiation, we chose two endothelial cell surface marker, CD144 and VGFR2. And over time, you can see the increasing expression of the CD144 and VGFR2 as a cell surface marker of endothelial cells. Cell viability, we didn't see any significant difference between different samples. During the culture, we studied the cells using light microscopy, and we noticed morphological changes. You can see in day 7, you have more spindle shaped morphology, and in day 21, dominated by cobblestone shape, which is PCs or endothelial cells. SEM scanning electron microscopy showed cell attachment to the surface of the RGB modified polymer in the A, and P is organ composite. In summary, we isolated endothelial progenitor cells and cultured them on composite samples. They remained highly viable and showed significant expansion over the time of experiment, and also increasing level of endothelial specific cell surface marker, revealed differentiation of the cells into endothelial cells. So we can conclude that this finding support the concept of in situ endothelialization, potential of the cardiovascular implants may be possibly similar composite polymer. However, we need to, our next plan is to do dynamic culture and also some vivo experiment to support this finding. Thank you very much.
Thanks. Good question. So the idea is to put this in vivo and if the progenitors latch onto it and cover it with endothelium. Is that
Yes, that's right.
So it's obviously a huge step from what you've shown where they've got nowhere else to go to actually assume that they'll attack endothelial progenitor cells in vivo and that they'll actually cover the substance.
Well, this was just proof of concept. Yes, just a start point. But yes, we want to do circuit pulsatile fluid to see the attachment to the valve, for example, from the circuit.
Well, just to follow up on that, you must have some data to show where they're seeded, because I mean, just showing adherence isn't the major factor here. It's adhering in the right place and how adherent it's going to be. So that's an important factor. And the other thing that I'm sure just before Banan chips in, are you confident in your market? Selecting, you truly, these are pretty, you did an enormous number of studies there. I just want you to answer that for him really before he asks the question. You always ask that question.
There's a sleep
To see the true attachment, we have to put the cells or the seeded valve into the suitcase to see how strong the attachment is. So the next step is to
you haven't got to that stage yet. Just showing that they're seeding.
Yes, and the proliferation and differentiation.
Yeah.
So following on from Professor Miller's question, your first histogram of your fact announcements, you showed there were many more double positive CD34s than CD123s. There were single positive cells. That's possible.
Yeah,
there must be something.
It's not the number, it's the frequency of the cells. I mean, we examine in a gated area, we examine just the frequency, the percentage of the expression, not the number of the cells. Perhaps the number is less than the single positive one, but the frequency is more.
What's wrong with the strategy of just coating them in vitro, which you can do, and then transplanting them or putting them where they need to go? Why do you want it to happen in
But the final idea is just to have a valve. Just after implantation, you can expect
That would be the ideal.
Yes.
I don't see what.
But you have to do some surface modification to attract the stem cells from the
Professor Bernard.
Just to show Professor Williams that I'm not asleep. Have you actually subcultured these cells and show that they're capable of growing independently subsequently? Was that they are colony stimulating?
Sorry,
When you've got them on your nanoparticle, have you then subcultured those and showed that they're capable of continuing to grow
subculture
the cells of the nanoparticle?
When we cultured the cell, the endothelial progenitor cells on the
Are
surface
they
of
growing
the
on colonies?
Yeah, we saw the colonies.
You clearly were asleep.